We'll continue on with the nervous system and take a look at our senses, and so our general sense of sensation as well as our special senses, and take a little bit bigger, deep look into those as well. In order to look at the sense of sensation itself, we need to take a little bit of a look at the receptors. And so the receptors themselves range in their complexity from just being things like a simple dendrite on a neuron to being things like actual receptors. They monitor both internal and external types of stimuli. And so they're looking at the different kinds of stimuli that are within the body, outside of the body. Some of it is conscious, some of it is unconscious information. As we come across a stimulus, depending upon how the stimulus is perceived, partially dictates on what that receptive field is. Is it a small stimulus or is it a large stimulus? And so this is going to help us to determine also where that stimulus itself is occurring. If it's a relatively small stimulus, it can make it a little bit more difficult to find, to determine exactly where they are at times. And if the process itself is relatively large, kind of over a larger area of the body, we may not be able to find it quite so easily either, depending upon the amount of stimulus that's there and determining where exactly we see it. Receptors themselves fire off at different rates. And so with some of our receptors, we have kind of a constant flow of information. And so we have tonic receptors. These are things like our proprioceptors. This is giving us information all the time about exactly where our body is at any given time. Versus things like phasic receptors. Phasic receptors are going to fire off based upon, in a sense, kind of a change in the situation. And so they tell us when things have changed from the last moment, which means that if we have constant stimulus, we're going to potentially have the situation where we kind of forget about it. We don't really necessarily have that constant stimulus coming in all the time. Things like jewelry, our glasses, a hat, even our clothing, if we don't move and kind of change the stimulus, the idea or the concept that it's there tends to start to fall away. And this is why you can have people that are walking around looking for their glasses, and all along their glasses were on top of their head. But because they hadn't moved, they were unable to actually sense them. For looking at the receptors, we have different kinds of receptors themselves, and we can look at them based upon kind of the basics here in terms of, are they special senses or are they general senses? And so receptors themselves are found in slightly different areas. General senses are basically all over the surface of the body. So these are related to our skin primarily, and so we have sensation there. The special senses essentially don't leave the head. So we have our vision, we have our smell, we have our taste, hearing, balance, are all up there in the head itself on there. Depending upon the type that we have, we can find them in different places as well. And so we can have exteroreceptors, and so extero kind of exterior, or we can have interoreceptors, interior kinds. And exteroreceptors are essentially the stimulus has come from outside of the body. And so it's either on the surface of our skin or it's one of our special senses. But either way, that stimulus came from outside of the body itself. Interoreceptors, the information is coming from inside the body. And so the stimulus itself is oftentimes within the viscera, so our organs. Some of it is conscious, some of it is going to be unconscious as far as that information goes. Proprioceptors go both ways as well, in that they are both conscious and unconscious, but the two of them mirror one another, and so we have essentially an unconscious knowledge of where exactly everything is on the body, as well as a conscious knowledge of where everything is as far as our joints and our muscles and our tendons and our ligaments go. We know exactly where those are at any given time. For the receptors themselves, we have different types of receptors based upon what they're going to respond to. And so we have things like chemoreceptors. They respond to some sort of chemical. These can be both intero and exteroreceptors, so smell and taste rely upon a chemical in a fluid in order to be able to detect it. We can determine changes in temperature, so we have thermoreceptors. This simply tells us a change in temperature, not exactly what the temperature itself is. We can determine that if we go from outside and it's hot to inside and it's cold, we can feel that difference that's there. Pain receptors or nociceptors, we can determine that damage to tissue is either occurring or is potentially going to occur. We're getting to the point where it's almost going to happen in a sense. Touch receptors tell us that something has touched the surface of the skin itself, and so these can be deep receptors. These for deep touch. These can be surface detectors for light touch. Again, our proprioceptive information gives us information about where our body is at any given point. And then pressure receptors. We have things like mechanoreceptors or baroreceptors telling us that something is pushing on something in a sense. For our special senses, we'll start off with taste. And with taste, we have the tongue here involved primarily. And on the surface of the tongue, we have what are called papillae. And so these papillae are kind of like structures that stick up off of the tongue. And on those papillae, we have the actual taste buds themselves. And so we have the actual structures that are going to respond to the chemical stimulus that are there. And so we have them all along the sides of those individual taste buds themselves. The different kinds we have filiform, we have fungiform, we have valate or circumvallate, and then we have foliate. Other than the filiform, all of them are involved in the actual sense of taste itself, and we'll talk about each one in just a moment. The circumvallate one here, the valate ones, make this kind of V shape at the back of the tongue. They're the least numerous of all of them. So filiform, they are kind of the most numerous as far as the actual taste buds themselves or the actual papillae goes, but they have no sensory function. They don't do anything for the sense of taste itself. Fungiform, we have the least amount of taste buds along each one, but they do function in taste. Valate or circumvallate, we have the least number of papillae, but they have a large number of actual taste buds on them. And so it kind of flips between the fungiform and the valate there. For our foliate ones, these ones are involved in typically childhood. This is where children's taste is constantly changing, and so they're predominantly used during childhood itself. And this is where one week a kid could enjoy their food, and the next week the exact same food, they don't really care for it. Or they start to try something new, and at first they don't like it, but then they eventually start to like it. And so that change is always occurring within the taste itself. When we think of taste, mostly we think of these four here: salty, sweet, sour, and bitter are our classical taste sensations themselves that are there. Salty is typically salts or metal ions, and so metals themselves. Sweet are organic compounds, things like our saccharides, so sugars, amino acids, things like that. We have sours, so these tend to be acids, and so they're more acidic in nature. And conversely, we have bitter, which tends to be alkaline. Alkaline things oftentimes are things that are potentially dangerous to us, so they typically are things that are potentially poisonous. The last one here, umami, is essentially the newest one of them all. This is kind of your sense of enjoyment in a sense, and so it has a pleasant taste to it. There's not really a distinct kind of a component to it. It is something that we enjoy, and so things like chicken soup give us that good feeling, so to speak, if you like chicken soup, or think of your favorite type of food. Umami would be the taste in a sense that being discriminated there for the sense of umami itself. Combining with the sense of taste, we have the sense of smell. And so when it comes to taste, primarily smell is the biggest component there, as taste really only gives us 5 different sensations. Smell gives us a large number of chemicals that we can actually detect and have distinct knowledge of each one of those. So smell is found way up at the top of the nasal cavity. That's where we have our olfactory mucosa that's there. So if an odorant, if a smell molecule makes its way down this way, we may not actually detect it. And so if there's very few of those actual chemicals, and it makes its way through the nasal cavity without getting up to the olfactory mucosa, we're not going to be able to sense that smell that's there. In the actual olfactory mucosa itself, we have different types of cells. And so we have the actual receptor cells, these little yellow guys here, with their processes extending down and then processes going up. So we have a bipolar cell. Those bipolar cells are detecting chemicals in solution inside the nasal cavity here. Next to them, we have supporting cells. And then lastly, we have our basal cells. The basal cells are stem cells. These are going to manufacture new olfactory cells, receptor cells, as well as supporting cells. So they're going to be constantly replacing the tissue that is there within the tissue itself that's there. Each side of the nasal cavity is independent of one another. So we have two sets of olfactory nerves, not just multiple nerves. They go up through the foramen in the cribriform plate to make their way up to the olfactory bulb, where they then synapse, and they synapse right away here, and they synapse on our next cells that then travel down through the olfactory tract and on their way out. For our sense of vision, we have, again, outside stimulus that comes in. The eyes themselves are going to house essentially the majority of all of our sensory receptors in the entire body. And so they have all of these different structures that are there on the back of the eye. So we need to protect them. So we have different processes that are going to help to protect the eyeball itself. And so we start with essentially a thin little covering that covers over the sclera as well as the eyelids. And this is called the conjunctiva. So it's a thin little membrane that's going to cover over the two. And it essentially seals off the eye socket from the outside world, so to speak. We then have things like our eyebrows, our eyelashes, our eyelids, all to help to stop anything from coming into the eye. So things like our eyebrows are meant to direct things like sweat away from the eye itself. The eyelashes are relatively sensitive to anything that touches them to allow them to close the eye really quickly as well. Up in this region up here, we have the lacrimal gland. That's going to secrete tears over the eye, which are going to make their way over to the medial side of the eye and get down here into the medial canthus. And there they're going to find openings to the nasolacrimal duct and allow them to exit and drain down into the nasal cavity. That's there. And so here we can see the conjunctiva. And so it surrounds the sclera up onto the eyelid itself as it covers over there. We have the eyelid or the palpebrae there as well, covering over. You remember back to when we talked about muscles, we talked about the orbicularis oculi muscle. And so this is for closing the eye itself and surrounds the eye. We also have 6 strap-like muscles that control eye movements, running on all 4 sides of the eyeball, as well as 2 oblique muscles that come in and help to rotate the eye as well. When we look at the eyeball, the first outer layer is our fibrous tunic. This fibrous tunic is the sclera primarily on the posterior side, and then the clear cornea on the front. Our vascular tunic is primarily made up of the choroid, and that again goes primarily posterior. And then we also have the ciliary body and the iris. And so we have 2 other structures that are there as well. The actual functional layer is the retina. And so we have all of the retinal material back here, giving us the sensation of vision itself. The eyeball itself, when we allow for light to come into the eye, it has to go through the cornea, through the pupil there, and then after it goes through the pupil, it's going to hit the lens. But sometimes with age, that lens, which is simply proteins, starts to become a little bit thicker. It starts to become a little bit opaque, and a person gets cataracts. And so cataracts can be fixed by replacing the lens inside. And the most advanced types of lens replacements actually allow for the person to have the ability to utilize the lens just like they would have a normal lens. And so they can see near, intermediate distance, and then far away as well without having to change any glasses or anything like that. As that retina comes back towards the back of the eyeball, all of the cells that are photoreceptors have sent their information over to what are called ganglion cells. And those ganglion cells are going to converge on the optic disc. The optic disc is our so-called blind spot. There are no visual receptors in this area. So since there's no visual receptors there, we don't have any kind of vision there. We can't see anything in that particular area. Where we have great amount of vision is in this area over here called the macula lutea. And so the macula lutea is our area where we have the light as it comes into the eye basically is focused on the macula lutea and the fovea centralis. And so we have the greatest amount of cones, very few rods in this area. So we have very great bright light vision. So we can see when it is bright out, but we're not going to be able to see as well when it is dark because we just don't have any rods there. One of the ways that we can see the optic disc or perceive that it is actually there is to utilize the little dot and plus mark here. And so if you were to look at the dot with your right eye and stare at the black dot there, and then look over, not really look over, but move your head in closer to the dot, you'll eventually find that the plus sign disappears. So using your right eye, stare at the black dot and move inward until the plus sign disappears. And that's because the light then falls on that optic disc as you're staring at the black dot. And so the brain fills it in with information that is kind of in the general area itself. So here we can see the macula lutea as essentially all of our cells move out of the way. And we're left with light to be able to come in here and strike right on the cones. And again, we have very few or no cones in that area, so we have very good central vision. With age, sometimes that macula starts to degenerate and we start to see changes there, and that can lead to blindness. And that's what this represents, is essentially what it would look like if you were looking with an eye that had macular degeneration. As that macula starts to become damaged, it then causes central blindness to occur. Within the eye, we can divide the eye up into different areas. And so the eye is divided up into a couple of different cavities. And so we have a posterior cavity. The posterior cavity basically goes from the back of the retina all the way to the lens. And so it contains this space here. The posterior cavity contains vitreous humor. And so vitreous humor is a jelly-like substance. We get it once as an embryo, and it sticks with us for the rest of our lives. The anterior cavity goes from the lens, I'm going to draw the line kind of in the middle of the lens, all the way up to the cornea. And so that anterior cavity is divided up. The anterior cavity then has space from the lens to the iris, and this is the posterior chamber of the anterior cavity. And then we also have from the iris to the cornea, we have the anterior chamber of the anterior cavity. That's there. And so it's going to be divided up into those couple of different places within. The anterior cavity contains aqueous humor. This aqueous humor is going to be constantly replaced. And so we're going to be always changing out that aqueous humor that's there, and it's being manufactured by parts of the ciliary body here. And so the ciliary body is producing this aqueous humor. It's going to come into the posterior chamber first, make its way out through the pupil into the anterior chamber, and then it's going to drain through these little openings here called the sclerovenous sinus or the canal of Schlemm. And so kind of two different areas there. So it's constantly being produced. Roughly every 90 minutes or so, we're replacing that aqueous humor that's there. Again, the vitreous humor that's in the posterior cavity here, this we got once, and it's going to stay there throughout our entire lives. If that fluid can't drain out of the anterior cavity and make its way through that canal of Schlemm there, the fluid's going to start to continue to build up, and pressure is going to build and build and build in this anterior cavity here. And as it builds in the anterior cavity, it pushes against the lens. The lens pushes against the gel of our vitreous humor, and then that starts to push against the retina as well as the optic disc, which will eventually lead to blindness. And so glaucoma is that buildup of pressure in the anterior part of the eye. This is where when you go to the eye doctor, sometimes they do the test with the puff of air. Other times they use little mechanical things where they numb the eye and then actually push on the eye to measure the amount of pressure that's there as well. When we receive information for vision, remember back to sensation, that information that comes through from the right side of the body ends up over on the left side of the cortex and vice versa. Vision is no different. And so that means that anything that is on this side of the body, on the right side of the body itself, that's there. This information needs to be then processed over on the left side of the brain. And so that information is going to cross over. But each eye basically sees a little bit of the right side and a little bit of the left side of our body. And so that means that each eye needs to send some information to the opposite side, and some of that information needs to stay on the same side of the body itself as well. And this is where the optic chiasm or optic chiasma comes into play. In this chiasm here, this is where we're going to cross information over. And so each eye is going to send information from one side of the body. So I'll use the same colors here. So this side is going to send its information over to the opposite side. So all of this red portion here gets over to the opposite side of the body there. And then all of the blue portion here needs to come over from the opposite side. But this eye also has a blue portion that stays on the same side. And so we're going to end up having a combination of information from each eye going to either side of the brain itself. And so the optic chiasm there is where we're going to determine that information that is there. It then heads through the optic tract, goes over to the thalamus, and then comes back to the brain via the optic radiations and gets that information back to the occipital lobe. The ear is involved in both hearing as well as equilibrium. So it has a role in two different processes for us. That's there. And so we can divide the ear up into different regions. And so we have the external ear, kind of everything that we can see on the outside, but the true external ear actually continues all the way in until the tympanic membrane. And so although we, in a sense, only see the actual ear structure, the auricle on the outside, it continues all the way in until the tympanic membrane. We then have the inner ear and the middle ear. The middle ear continues on from the tympanic membrane over to the oval and round windows. The inner ear then is deeply embedded into the temporal bone, and it's going to have all of its structures inside the bone itself. And so we can then determine out both hearing and equilibrium. So the outer ear does only hearing, the middle ear does only hearing, and the inner ear does both hearing and equilibrium. So our external ear begins with the actual cartilaginous ear that we can see here. And so we have different structures that we can see on the ear itself. We have the actual auricle, so the actual outer rim of the ear itself, that's there. Things like our helix and antihelix and tragus and antitragus are essentially been cut off on the ear here itself. So we can't actually see those structures on this particular image. But we can extend through the external auditory canal up to the tympanic membrane. And the job here is to basically funnel the sound into the ear. Think about if you can't hear something that's kind of distant, you usually kind of cup your ear to make it a little bit larger to allow you to gather more sound. When we look in through that external auditory canal, we can see the tympanic membrane. And so we can see a nice representation of the tympanic membrane there, nice and healthy. It's not quite transparent, but not quite translucent either. So we can see a little bit through it, but it's not a perfect image, so to speak, as we see the structures come through there. The middle ear, because it is air-filled and because it does have a connection down to the nasal cavity, and we'll talk about that in just a minute, it has the potential for infection. So here now we can see that there's kind of a greenish hue behind the tympanic membrane. We've got a middle ear infection going on there. That middle ear infection here has become very prolific. Lots of redness out into the ear canal itself. The tympanic membrane is bulging out of its space. And if it continues to be untreated, it can actually rupture. And so it can actually rupture the tympanic membrane. Now we can see all of our ossicles below. We can see the malleus, the incus, and the stapes behind there. You can see that it is a mucous membrane, so it's nice and shiny when we shine a light in there because it's moist. So the middle ear begins at that tympanic membrane, and it's going to continue all the way in until the oval and round windows. And so we see the boundaries there for that structure. It contains the malleus, the incus, and the stapes. And so we have three separate bones there, the smallest bones in our body. The malleus actually connects to the tympanic membrane here, and then the malleus to the incus to the stapes, and then the stapes butts right up against the oval window. Both the malleus and the stapes have a muscle associated with them. And so the stapes has an easy one called the stapedius. And so it attaches into the stapes there. And then we also have the tensor tympani. And so the tensor tympani is going to connect up to the malleus and actually continues down through the auditory tube or the eustachian tube. And so we see that continuing down through that tube in the middle ear. The eustachian tube itself is what's going to connect us to the nasal cavity. This is what's going to allow us to balance the pressure inside the eardrum there. And so this is where when you're going in a plane or a tall building and take the fast elevator, your ears start to pop because the air pressure is either going to go into the ear, we're going to get air coming in to equalize the pressure, or we're going to get air going out to equalize the pressure depending upon whether the pressure has increased or decreased around us. So here we can see the actual eustachian tube or auditory tube as it travels up from the nasal cavity there. Usually it is closed, but it does open when we do things like chew or yawn or swallow. That all kind of opens it up and allows us to equalize that pressure at any given time. Most of the time throughout the day, we don't even notice it. It's those times when pressure changes quickly, like in a plane or in an elevator, where we then notice popping. In some people, they get infections quite often in their middle ear, and one of the things to fix it is to put in ET tubes. And so these tubes essentially create a small opening in the tympanic membrane. That opening in the tympanic membrane is then held open with the actual tube itself and allows for the person to utilize that tube rather than the eustachian tube. And so they no longer have to rely upon any kind of movement going up from the nasal cavity, and that hopefully allows them to grow enough and get out of that process of having constant middle ear infections. And so as we get to the inner ear, we now have the ability to do our actual detection of hearing and balance. The cochlea is going to take care of hearing. The semicircular canals and the vestibule are going to take care of balance. And so we're going to see those in those particular areas there. They're both innervated by the vestibulocochlear nerve, which then splits and gives off portions coming to the cochlea and then over to the semicircular canals and vestibule, so that we can see the structures that are there. The inner ear here itself has kind of two different portions to it. We have what we call the bony labyrinth and the membranous labyrinth. And so the bony labyrinth is essentially the portion within the temporal bone itself. And so that portion that is outside here within the bone. And then the membranous labyrinth is the portion that makes up the actual structures themselves inside. And so where we have our semicircular canals and where we have the cochlea. So on this picture, it's kind of the pink areas is the membranous labyrinth that's there. Inside the vestibule, we have a couple of different structures to look out for. We have the saccule there, and then we have the utricle, and then we have an ampulla for each of the three semicircular canals. Each of those three semicircular canals lies in each one of the planes of directions, and so each of the X, Y, and Z planes, so that we can monitor as we move around throughout our day. Inside the vestibule, we have essentially a series of hair cells that are embedded into a membranous gelatinous membrane. Inside that membrane are little otoliths, little tiny stones. And so these little tiny stones add for weight, so that as we tilt our head forwards and backwards and side to side, that pushes and pulls on the membrane, which then pulls our hair cells, and we detect that we have moved. For our static equilibrium, we use those otolithic membranes, and that's just simply tilting our head forwards and backwards and side to side. For dynamic equilibrium, for rotational type of equilibrium, that's where we have our semicircular canals. And so these semicircular canals themselves are there to monitor when we are essentially spinning. And so there's fluid inside of them, and that fluid's going to spin around and move, and then it's going to bump into this little cupula here. As it does so, as that fluid pushes along there, it's going to bend that structure over, and once again we have hair cells that are going to detect that movement. Each ear is going to have essentially a different rotation. So if you were sitting in like a office chair desk and spun yourself around, that semicircular canal would spin the fluid inside of it, and our little cupula here would detect that we're spinning. One ear would detect rotation in one direction, the other ear would then detect the rotation in the opposite direction. And as long as the two ears kind of agree in terms of the rotation, then we're going to have good balance. But if that information gets kind of disrupted, particularly because in one of the ears we have a otolith that has become dislodged, then we're going to have movement in one ear that's either greater or even less than the other side, and that's going to lead to vertigo for that person. And what this little demonstration here shows is a maneuver to remove that otolith out of the semicircular canal and get it back into the vestibule, so that the person can then move around throughout their day without any kinds of problems. If they can't get it out with this maneuver, then they may have to go in and do surgery to do it. Typically, they only get 1 or 2 chances to try and with this maneuver before the person is extremely nauseous and does not want to continue with the process itself.